So, welcome to today's lecture. As we have discussed in the last lecture, we are going to discuss today about flow cytometry. Okay. So, flow cytometry. So, from the name you can understand that there are some flow of liquid, right, and cyto means cell and metry means measurement. So, cell in suspension or in fluid, we are going to measure the some parameters of cell. That is why the techniques is called flow cytometry, and the instrument we are going to use is flow cytometer. Okay. So, this machine is also very commonly known as FACS, fluorescence activated cell sorter, but this is not the name of the general name of the machine. FACS is the name of a company. Okay. The Becton Dickinson named it. So, what actually doing it? By this, as we have discussed in the last class, if I stain a cell with a fluorescence dye that and see under the microscope, we can see few cells, maybe 100 if you give a lot of time. But if you want to measure in population study, like what is happening in 10,000 cells or a million cells, then manually it is not possible. So, this machine is actually going to help you how many cells are what color, and that color depends on what you want to do. So, by this machine what we can do is we can measure the size, or we can differentiate a mixture of cells of different size and their complexity, and definitely the protein expression. If we would like to determine any protein, that we can use the same immunostaining method by fluorescence labeled antibody, and we can measure it. Okay. So, there are three basic components in this machine. One is definitely we have to handle some liquid. So, that is fluidics. Okay. So, that needs a pump which will suck the cell and do the pass the cell through the detector. And second is we just need, like any other spectrophotometer or any measurement, because we are going to use the light to see the fluorescence and other parameter, we need optics. And third component is just to measure and to digitize the information that we are getting is electronics. Okay. So, this is three components are very distinctly doing their own job. So, fluidics, optics and electronics. What is there? The basic unit is, so there is a light source. Okay. So, light is going in the path of the light. If there is a cell, suppose this is the nucleus, and this light will interact with this cell, and there is a detector. This is a common what I am saying. There is a detector here which will detect the signal and give it. So, cell is going to come here by fluidics, optics is doing its job, and the detector which is linked with a photomultiplier tube PMT, photomultiplier tube, which can amplify the signal if you need it, and then if this signal will go and electronics part will get the information and digitize the information. Okay. So, this machine actually is completely computer operated, and so you do not have to do anything with the machine. Machine has very simple button. There are only 6 buttons, 3 buttons actually controlling the flow, okay, how fast the cell will move. So, there are 3, one is slow, medium and fast. Another button is run. So, when you are ready, you press the run, it will do its own job. So, when the run is complete, third thing is standby. It will automatically go to standby, but if you want to do it forcefully, like okay, something is wrong or you do not want anymore, you want to stop the calculation or the measurement, you press the standby. And the sixth one is called prime, that is normally we do not use, just to clean the machine or the pipe. Okay. So, what is there actually? So, let me go stepwise. So, if you take, suppose this is a tube, okay, and there are cells. Okay. There are cells. So, if you take blood, what is there? Blood is composed of varieties of cells, okay. Different size, different, blood has platelet, blood has RBC, blood has lymphocyte, monocytes or macrophage, neutrophil, basophil, eosinophil, all we have discussed. So, it has varieties of cell here. Okay. I hope many of you know, or all of you know, the bacterial cell counter or the Coulter counter, the electronic counter. What is there? In bacterial counter, the idea is very simple. So, you take bacteria in a tube. So, there are a lot of bacteria here. So, and if you have a capillary here, so and then you suck the cell. So, what will happen? Bacteria will move from solution through this pipe, right. And if there is a light source here, and what is going to happen? Suppose light, and there is a photomultiplier tube. I am taking the simplest way of measurement. What is, as soon as one cell will come here, there will be a shadow, and that shadow will be counted, and that is very fast. You do not have to count by hemocytometer, and you do not have to pay. Just put that machine, run it. So, each cell will go, each shadow will be counted, and finally, you can say after certain time or in 1 ml or 10 microliter or 100 microliter how many cells are there. So, that is how bacterial counter works. It is very, this idea, if you expand and do lot of good, more activity, then these facts will come. It also, you take the tube, you have a very thin capillary. Okay. So, there is a suction, cell will move through this capillary and move through this capillary this way. What happened? There is a tank. In this tank, there are certain fluid. Okay. This is called sheath fluid, which is mostly, this is a commercial trade secret. It is not known exactly. Normally, the company is supplying it, but it is generally the phosphate buffered saline. So, that cell, you have to maintain the osmolarity. So, cell should not lyse. Okay. So, from that source, what happened? There is a pipeline. I am not going detail of this drawing. What is happening? Up to certain level, there is a capillary, what you can see, and after that, inside the machine, through which cell will move, okay, upward. After that, within the machine, there is a flow of liquid. So, automatically from both sides, if the liquid flow like this, so there will be a jet, and through this cell will move blank. There is no pipe or anything. So, while this cell is going, light is coming from this side. Say this case, the light is laser. So, light will pass, and there is detector. That detector will detect. What this detector will detect? First thing what I will say is, it is called forward scattering. Okay. Forward scattering, and in short it is called FSC. So, what is happening? So, suppose this is the cell, this is nucleus, light is coming like this, light coming this way. So, now if you see, this cell will block some light and this will go. If I make it over simple, just to make you understand easy way or easier way. So, what I am saying, so then this there will be a, so suppose there is a plate here, what will happen? There will be a shadow. Okay. Shadow is this size, right. So, if there is another cell which is say small, okay, which is small. So, same cell, similar cell, but this is a small cell. Okay. Same way, and if the light comes, suppose this cell is not there, this cell is there. So, I am drawing parallelly, but it will happen one by one. So, again same source will give the light. What will happen? The shadow size will be smaller than this. So, if there is a detector which can detect the area of shadow, what will happen, and I am going to plot them. I am going to plot according to size. So, what you will see, you will not see the cell. Okay. What you will see a graphical representation of this. If this is the size and this is arbitrary unit, if this is the size, each cell will pass that capillary, you will get one dot. Suppose the size is increasing this direction. So, what will happen? Suppose there is a big cell, you will get a dot here. If there is a small cell, you will get a dot here. So, suppose this is the smallest cell and this is the biggest cell. Okay. So, all the cells will be either this size or in between this size. As a result, so if you run 10,000 cells, you will get 10,000 dot. Where the dot will be? So, dot will be like this. Okay. Each cell will go, you will see a dot. Okay. So, that means in this region you will get lot of dots. So, this region will be the smallest one, this region will be the biggest one. Clear? So, forward scattering wise, each dot is going to tell you how many cells are big, how many cells are small, how many cells are in the medium size or in between small and big. Same way, there is another parameter we can measure. This is called forward side scattering. The light will scatter sidewise. Okay? Side scattering. What is going to happen if this is the cell? If this is the cell and this is say nucleus, again light will come same direction, same okay. But what will happen? Some light which is interacting with the cell will go, and if there is a particle inside, that will deflect. Okay. It will deflect this way. Variety, if there is no particle, it will go away, and if there is some particle here, there are a lot of particle, it may be nucleus, and it will deflect in different directions. So, all this side wise deflection of the light will be measured, or sidewise scattered light will be measured. So, which cell will scatter sidewise most? If they have more particle inside. If cell does not have more particle, most of the light will go. Okay. So, if you remember the blood, what is there? There are neutrophil. If you remember the neutrophil, sorry. If you remember the neutrophil is complicated nucleus. It has lobe like structure, right. You will see much better picture in book, and there are a lot of particle inside. And if you remember the mast cell, there are a lot of histamine granule inside. So, there are a lot of particle. If you see lymphocyte, there are very little particle inside, right. Macrophage, bigger, but less particle. Okay. So, now if I see neutrophil, okay, neutrophil is what? Neutrophil is particle wise. If I say that this inside is complex, okay, the granularity is much more. So, neutrophil is complex as well as big. Macrophage is not much complex, medium complexity, okay, but big. Lymphocyte is small, sorry, small should not be here. Lymphocyte is not complex at all and small. So, what is the combination? So, it is not medium complexity, complex, medium complexity, or I can say high complexity, medium complexity, or low or no complexity. Okay. Low or no complexity. Low complexity. So, high complexity is the neutrophil, medium complexity is macrophage, low complexity is lymphocyte. Size, they are big, this is big, this is small. Now, if I make a plot according to this, so again I mean I am coming back to this side scattering. So, if I plot this, and this is the complexity or granularity. Okay. So, this is side scattering. So, side scattering is increasing means complexity is increasing. Complexity is increasing. So, more complex cell is neutrophil. So, neutrophil will come here, macrophage will come here and lymphocyte will come here. So, you will get very 3 distinct population. Okay. All dot means each cell, normally we call it event. Okay. So, 3 different complexity, high complex will be here, medium will be here, low will be here, because it is distributed according to the internal granularity or the complexity. Now, if I draw the same, and one good thing I should mention that this particular machine, when one cell is going upward, all the parameters are going to be measured, because you will not get this cell, you will not get this cell back. So, if you remember this part, what is happening, cell will go through this liquid from this tank and it will be collected in another tank, which is waste tank. Okay. Waste tank means that all cell will be mixed, you cannot get that back, it will be too diluted, you cannot use it, that is one disadvantage of this machine. But once it is going, so it has several source of light and it has a several detector. So, it has a detector for SSC, it has a detector for FSC. Okay. I will come some more detector. So, for the timing we have 2 detectors. So, one cell will pass here, SSC will be measured, same cell will go here, FSC will be measured. So, size complexity will be measured of the same cell simultaneously. So, data is stored in the computer, clear. So, now if we data is already there. So, now if we plot, suppose this is size means forward scattering and this is the complexity. Complexity means side scattering. So, if I ask the computer to plot this, what is going to happen? I am going to plot this, because cell has this. I am not considering the RBC and platelets, because you can remove it, platelets are too small. Okay. Because when you measure something, your machine has certain limit, it cannot make measure something bigger than a limit and it cannot measure if anything too small than its limit. But in blood cell, lymphocyte to neutrophil, they can measure very nicely. So, what is going to happen? Size wise, if I distribute, what will come first? It will give a population here. Okay. So, this is what? So, if I see this is less complex, smaller in size, less complex, because this is here, complexity is increasing this way, and smaller size. Then medium size is what? Medium size is macrophage. So, macrophage will come here and it is little big. So, it is come here and complexity is medium. So, it is slight more complex, if this is the complexity scale. So, it is slightly more complex than lymphocyte, right. Now, if I go for neutrophil, it is bigger, not as big, I mean too big, but it is very close to macrophage, but bigger than macrophage, but highly complex. So, it will be slightly bigger than macrophage is here, this is lymphocyte. If it is slightly bigger, it will come here, but is highly complex. So, it will come here. Okay. And there are some in between complexity, it will come here. So, that way if you just run the blood cell, automatically this machine will distribute according to their complexity and size. So, you do not have to do anything extra, you take the particular cell you want to measure, suspend it, it should be single cell suspension. If it is a clump, you cannot do that. Okay. And you can instruct the machine through computer, I need count of 10,000 cells or I need count of 1,000,000 cells. Okay. So, that I am not going experimental detail because there is no point, you just understand the principle, that should be enough for this. So, okay. So, this is the distribution plot, this is called dot plot. Okay. This is called dot plot, but if I want to plot a histogram plot of this, suppose this one, this 3 population 1, 2, 3. What is this? This is, suppose this is 1, this is 2 and this is 3. So, this 1, 2, 3 will come 1, 2 and 3 in this plot where only complexity and this is arbitrary unit is measuring. Okay. So, now same plot we can also ask the machine to make a histogram. How it will come? It will come like that area is going to give you, that area is going to tell you that what this peak are. So, this is again this is same thing, this is 1, that means this is population is neutrophil as lymphocyte, this is macrophage and this is neutrophil. So, you can have dot plot, you can have histogram plot, you can have contour plot, okay, density plot, whatever kind of plot you want, you just have to click in the right place in the computer software or the program. You do not have to do, but cell will go and all data is stored. Now, it is up to you which way you want to count. And another good part of this machine or the software or the program is, if you just get this region or select this region by mouse, okay, and ask, suppose this is 1, 2 and 3, 1, 2 and 3 and ask that give me statistics. It will immediately tell you how many cells are there in the region you selected for 1, how many cells are there in region 2, how many cells are there in region 3. So, you do not have to do anything, you just take the cell in a tube, fit into it, press run, it will go automatically, everything will be done, you just get the data from computer and do it, I mean analyze it depending on what you want to do. Okay. But this is not as simple as this, definitely you need to calibrate the machine, you have to know the software. So, this part is without fluorescence. Same way what we can do is, suppose we have in blood in lymphocyte, say in lymphocytes, what we have? We have a general population of T cell, we have B cell, right. So, let me consider only this, there are many, I mean there are NK cell also. In T cell also we have T helper cells, we have cytotoxic T cell and we know what is their marker. T helper cell has what? CD4, not one, they have many CD4. Okay. Similarly, cytotoxic T cell has what? Cytotoxic T cell has CD8. Okay. What I did not tell you, B cell has another kind of marker, it is CD19. So, it is okay. CD19 is there. So, these see all are CD8. So, now the technique we learned before is what? Immunostaining. What we can buy is, we can buy anti CD4 antibody, I can buy from market, which is labeled with a red fluorescence. Same way I can buy anti CD8 antibody, which is green labeled. Okay. So, now if I take blood, where all the lymphocytes are there, or if I can take cells from spleen which is loaded with B cell and T cell, not blood say. I isolate the spleen and from spleen I isolate the cells. So, that will be loaded with B cell and T cell. Okay. So, all B cell and T cell will be looking same. So, you cannot distinguish which one is what. Okay. So, it is mixture, and there are a lot of other cells also. Okay. All are, say you isolate the lymphocyte, NK cell is also there. So, now here in this cell mixture, if you add this, the antibody, what will happen? Antibody will go and bind here, which is green. So, under microscope or fluorescence microscope, what we will see is some cell you will see green. After that, you wash the extra color, then you add this one. In the same sample, what will happen? All the CD4 cell will be red, because if this is the cell, if suppose this is the cell, what will happen? All antibody will go and bind, right? All antibody will go and bind on the receptor, because this is the antigen, and each antibody, suppose I am talking this one, if antibody is red, okay, antibody is red because you purchase red color. So, if you see this cell under microscope, cell will be look like this red. Okay. So, some cell will be red, some cell will be green, some cell will be colorless. Clear? Now, you remember this. So, what we have to remember? We have to remember that CD8 is green, CD8 is green, CD4 is red. We have to remember that. Okay. So, when I am talking about the light source and detector, so what is there? Here also in that laser, there are specific laser which can activate the red fluorescence. Okay. So, and cell is here, this is a cell, and there is a detector which can detect red fluorescence. Similarly, another light source is there which can activate the green fluorescence. So, cell is there, if it is green, detector is green. Okay. So, in this case, if this is, suppose this is the light source, this is for red, this is the light source which is green, and you have cell here. Okay. Cell, from where it is coming? It is coming from here. Okay, it is coming from here. Or if I go like this, if the cell is moving this way and the light source are here, this is red, this is green, detector are here. So, this is green detector, this is red detector. Okay. So, when the green cell will go here, when the green cell will come here, same way we plot. What is the plot? So, here we will divide this plot here. Here we will divide the plot this way. Anything beyond this line is green. Okay. Anything beyond this line is green, and this is colorless. So, when one green cell will come, one dot will come here. When red cell will come here, so this will come here. So, any green cell is going to come here, all other cell will come here. So, same way you will get lot of dot here for colorless cell and lot of dot here for green cell. Same thing we can get, for red. So, when the red cell will go, So, cell will go one by one, right. So, one cell will red, then some colorless cell will come in between, then some green cell will come. So, it is random. So, when this will pass, one red cell will go, one dot will come in the red, one dot will come in the red zone, and one green cell will come, one dot will come in the green zone. So, you can see simultaneously. So, we will get lot of red cell here and lot of colorless cell here. So, in that way, if you mix the cell and measure it and ask for the statistics, what we can get? We can tell how many red cells are here. What is the red cell? What is red cell? Red cell means CD4, right. So, we can easily tell how many CD4 cells are there. Same way, I can tell how many green cells are there. What are the green cells? Green cells is CD8. So, in a population within say 30 seconds to 1 minute, we can tell how many, what is the percentage, because total cell I know. I can tell you what is the percentage of CD4, what is the percentage of CD8. Okay? That way you can count any kind of blood cells if you know the marker. So, that is actually facts. Our flow cytometry, what we can measure also this way, like this. So, red is this, total is this, or vice versa. Okay, so, this is the flow cytometry, and in next lecture or next class, whenever we need flow cytometry, then you can understand it little more or little better way than what you understand, and I will suggest you to go and read the book. Okay. Because this is, you do not have to be master in flow cytometry, but you should understand what is this. Till then, see you in the next class.